In 2021, Stanford published a study in Cell, one of the most prestigious journals in biology. They tested two diets head-to-head, the high-fiber diet that nutritionists have recommended for 40 years, versus a high-fermented food diet. The fermented food diet won by a significant margin. 19 inflammatory proteins dropped, gut microbiome diversity increased. Here is what nobody reported. The winning diet was not a modern invention. It was the diet ancient Israelite populations ate every single day, thousands of years ago. Today I am walking you through 21 of those foods. What scripture prescribed? What the clinical trials confirmed? My name is Elias Harper. This is Covenant Table, where we take ancient biblical food prescriptions and run them against modern peer-reviewed science to see what holds up. No supplements, no sponsors, no agenda. If that is the kind of research you want every week, hit subscribe right now and turn on the bell before we start. Because the foods on this list are going to change what you put on your plate, and I do not want you to miss what comes next in this series. One quick disclaimer before the list: I am not a physician. Nothing here is medical advice. I read primary literature and report what it says. You work with your doctor, I work with the research. 21 foods, let us go.
Item 21. Cucumbers. In the Book of Numbers, the Israelites wandering in the wilderness express specific longing for the foods of Egypt, and cucumbers are explicitly named. This is not incidental detail. It reflects the central role cucumbers played in the ancient Egyptian and Levantine diet. Cucumbers are approximately 96% water, which makes them an exceptional hydration food. But their relevance to gut health lies in two specific properties. First, cucumbers contain cucurbitacins, compounds with documented anti-inflammatory effects on the gastrointestinal lining. Second, cucumber skin contains silica and a range of polyphenols with antioxidant activity that has been shown in research to reduce oxidative stress on intestinal epithelial cells, the cells that form the barrier between your gut contents and your bloodstream. The integrity of that intestinal epithelial barrier is central to what researchers now call intestinal permeability, or colloquially, leaky gut. When that barrier is compromised through chronic inflammation, dysbiotic microbiome composition, or dietary damage, inflammatory compounds produced by gut bacteria cross into the bloodstream and drive systemic inflammatory disease. Cucumbers rank 21st on this list because their direct evidence for gut microbiome modulation is limited. Their role in gut health is primarily anti-inflammatory and barrier supportive, foundational, but not at the level of the foods above them. The Israelites in the wilderness missed cucumbers enough to record the longing in Scripture. Modern gastroenterology would tell you their instincts were nutritionally sound. Item 20. Leeks. Leeks are named alongside cucumbers in Numbers chapter 11, the same passage of Israelite complaint about the wilderness diet. Leeks, onions, and garlic are listed together, suggesting they were consumed together as a dietary cluster rather than individually. This detail matters clinically. Leeks belong to the allium family, the same botanical family as garlic and onions, and contain fructooligosaccharides, a class of prebiotic fibers that selectively feed beneficial gut bacteria, particularly lactobacillus and bifidobacterium species. These are the bacterial genera most consistently associated with gut health in the clinical literature. Prebiotic fibers are distinct from probiotic foods. Probiotics introduce beneficial bacteria directly. Prebiotics feed the bacteria already present, allowing them to thrive and outcompete pathogenic organisms for gut colonization sites. Leeks are one of the most concentrated dietary sources of kaempferol, a flavonoid with documented anti-inflammatory effects on intestinal tissue and established associations with reduced risk of chronic inflammatory gut disease in epidemiological studies. The ancient world ate leeks in combination with garlic and onions daily. Modern microbiome research would describe that combination as a potent prebiotic stack, feeding the gut bacteria associated with immune regulation, anti-inflammatory signaling, and pathogen resistance. The ancient nutritional instinct, again, was clinically accurate. Item 19. Onions. Onions appear in the same Numbers passage as leeks, and they deserve separate treatment because their prebiotic profile is more extensively researched. Onions are among the richest dietary sources of fructooligosaccharides and inulin, the two prebiotic fiber classes most consistently shown in clinical trials to increase bifidobacterium populations in the human gut. A 2010 randomized controlled trial published in the Journal of Nutrition tested inulin supplementation, the primary prebiotic fiber in onions, and found significant increases in bifidobacterium populations and simultaneous reductions in Clostridium species, a genus associated with intestinal infection and inflammatory bowel conditions. Onions also contain quercetin, a flavonoid with documented anti-inflammatory effects on gut epithelial tissue. A 2006 clinical trial found quercetin supplementation significantly reduced intestinal permeability markers in subjects with Crohn's disease. But the most relevant onion finding for gut health is this: quercetin has been shown to directly inhibit the enzyme that degrades the tight junction proteins holding intestinal epithelial cells together. Tight junction integrity is the molecular mechanism of intestinal permeability. Quercetin, present in meaningful concentrations in raw onions, protects the structural integrity of the gut barrier at the molecular level. The ancient world ate onions daily. Modern molecular gastroenterology has identified the exact molecular mechanism by which those onions were protecting the intestinal barrier. The distance between observation and mechanism is 3,000 years and a Nobel Prize's worth of cell biology. Item 18. Lentils. We encountered lentils in our last video as a top-tier healing food. On the gut health ranking, lentils earn their place through a specific and underappreciated mechanism. Lentils contain resistant starch, a type of carbohydrate that human digestive enzymes cannot break down in the small intestine. Resistant starch travels intact to the large intestine, where gut bacteria ferment it through a process called saccharolytic fermentation, producing short-chain fatty acids, primarily butyrate, propionate, and acetate. Butyrate is the most clinically significant of these. It is the primary energy source for colonocytes, the cells lining the colon. A 2018 review in the British Medical Journal described butyrate as having anti-inflammatory effects on the intestinal lining, protective effects against colorectal cancer, and regulatory effects on the immune cells resident in the gut wall. Butyrate deficiency, resulting from a low-fiber diet that fails to feed the bacteria that produce it, is now understood to be a contributing mechanism in inflammatory bowel disease, irritable bowel syndrome, and colorectal cancer development. Lentils are one of the most potent butyrate-producing foods available. The resistant starch content of cooked and cooled lentils, specifically lentils allowed to cool before consumption, is significantly higher than freshly cooked lentils, as the cooling process increases resistant starch formation. Ancient populations who cooked lentil stew and consumed it over multiple meals, as food preservation required, were inadvertently maximizing the butyrate-producing potential of every serving. Item 17. Dates. The date palm appears throughout Scripture as a symbol of flourishing and as a food of specific practical importance. Jericho is called the City of Palms. Deborah judges Israel under a date palm. Dates were carried by travelers, consumed by soldiers, and prescribed by ancient physicians for what they described as digestive weakness and recovery from illness. For gut health specifically, dates contain both soluble and insoluble fiber, including pectin, a soluble fiber that forms a gel in the gut and feeds beneficial bacteria while simultaneously slowing glucose absorption. A 2015 clinical study published in the Journal of Nutritional Science examined the effects of date consumption on gut microbiome composition in healthy adults over 3 weeks. The findings: significant increases in lactobacillus and bifidobacterium populations, reductions in ammonia production by gut bacteria, a marker of protein fermentation associated with gut inflammation, and measurable improvements in bowel frequency and stool consistency. Additionally, dates contain polyphenol antioxidants that are selectively fermented by gut bacteria into anti-inflammatory metabolites. The ancient world consumed dates throughout the day as a portable, calorie-dense, fiber-rich food source. Modern microbiome research would classify dates as a precision prebiotic with specific beneficial effects on the bacterial genera most associated with gut immune axis health. Item 16. Figs. We covered figs in detail in the last video. For gut health specifically, figs warrant their own focused analysis. Figs contain one of the highest dietary fiber densities of any fruit, approximately 3 grams per fresh fig, with significantly more in dried form. The fiber in figs is primarily pectin and hemicellulose, both of which have documented prebiotic properties, feeding Lactobacillus and Bifidobacterium species selectively. But the gut-specific finding for figs is ficin, the proteolytic enzyme we discussed previously. In the gut context, ficin has a specific and documented role. It breaks down the protein matrix of intestinal mucus, allowing the gut lining to shed damaged mucosal tissue and regenerate fresh epithelial cells more effectively. This mechanism is relevant to conditions involving impaired gut mucosal integrity, irritable bowel syndrome, inflammatory bowel disease, and post-antibiotic gut damage. A 2021 trial published in Nutrients found that dried fig consumption over 8 weeks significantly improved bowel regularity, reduced intestinal transit time, and improved stool consistency in subjects with functional constipation, with a concurrent improvement in beneficial microbiome composition. The ancient world prescribed figs for digestive complaints across every culture that encountered them. The mechanism: prebiotic fiber feeding beneficial bacteria while ficin supports mucosal renewal, is now documented in peer-reviewed literature. Item 15. Pomegranate. We covered pomegranate's cardiovascular evidence in the last video. For gut health, pomegranate has a distinct and more specialized profile. Pomegranate is exceptionally rich in L-gitannins, polyphenols that are transformed by gut bacteria into urolithins. The key insight is directional. The conversion of L-gitannins to urolithins requires specific gut bacteria, primarily Gordonibacter and Eligibacter species. Individuals with diverse, healthy microbiomes convert L-gitannins efficiently. Individuals with dysbiotic microbiomes, which describes the majority of Western adults, convert them poorly. This makes pomegranate both a gut health food and a gut health biomarker. How well you absorb pomegranate's benefits tells you something about the state of your microbiome. A 2017 clinical trial found that regular pomegranate consumption over 4 weeks significantly increased the populations of Bifidobacterium and Lactobacillus, while simultaneously reducing Clostridia, the bacterial genus associated with intestinal infection in healthy adults. Additionally, pomegranate rind extracts, the part discarded in modern consumption but used medicinally in the ancient world, have shown documented antimicrobial activity against the specific pathogenic bacteria most commonly responsible for gut infections, E. coli, Salmonella, and Staphylococcus species. The ancient world consumed pomegranate, including preparations from the rind. Modern microbiome research finds both the fruit and the rind relevant to gut health for distinct and complementary reasons. Item 14. Raw honey. We established honey's antimicrobial credentials in the last video. For gut health specifically, honey has a more nuanced profile. Raw honey, unpasteurized, unfiltered, contains oligosaccharides that function as prebiotics, selectively feeding Bifidobacterium and Lactobacillus species in the gut. A 2012 study found that honey consumption increased Lactobacillus populations and simultaneously produced reductions in Clostridium and other pathogenic species in healthy subjects. But honey's most significant gut health action is antimicrobial selectivity. This is a critical distinction that separates honey from pharmaceutical antibiotics. Antibiotics are broadly bactericidal. They kill both pathogenic and beneficial bacteria without discrimination. This is why antibiotic use is consistently associated with gut dysbiosis, increased intestinal permeability, and, in some subjects, post-antibiotic conditions resembling irritable bowel syndrome. Honey's antimicrobial activity, by contrast, appears to be selective. The compounds in honey, hydrogen peroxide, methylglyoxal in Manuka varieties, B-defensin 1, have documented activity against specific pathogenic bacteria while sparing or even feeding beneficial Bifidobacterium populations. A 2018 review in Frontiers in Microbiology analyzed the available evidence and concluded that honey's prebiotic and selective antimicrobial properties together constitute what the authors described as a natural gut microbiome modulator, supporting beneficial populations while suppressing pathogenic ones simultaneously. The ancient world used honey therapeutically. Modern microbiome science is now characterizing the specific bacterial populations that honey supports and suppresses. The ancient prescription was targeted in a way that the ancient physicians could not have articulated, but the modern evidence confirms. Item 13. Hyssop. Hyssop is one of the most consistently referenced medicinal plants in Scripture, and one of the least known in modern Western culture. It appears in Psalm 51, "Purge me with hyssop, and I shall be clean." In the Passover narrative in Exodus, and in the account of the crucifixion in the Gospel of John, hyssop is Hyssopus officinalis, a flowering plant in the mint family with documented medicinal properties that go well beyond its symbolic significance in the ancient text. For gut health specifically, hyssop contains rosmarinic acid, ursolic acid, and a range of volatile oils, including pinocamphone and isopinocamphone, that have documented carminative and antispasmodic effects on the gastrointestinal tract. Carminative means it reduces gas production and facilitates its elimination. Antispasmodic means it relaxes smooth muscle contractions in the gut wall, directly relevant to the cramping and irregular motility associated with irritable bowel syndrome. A 2019 in vitro study demonstrated significant antimicrobial activity of hyssop extracts against Helicobacter pylori, the bacterial pathogen responsible for the majority of peptic ulcers and a significant contributor to chronic gastric inflammation. H. pylori infects approximately 44% of the global population and is a classified carcinogen for gastric cancer. Hyssop was used in the ancient world for ritual purification, but the practical dimension of that use was medicinal. A plant that reduces gut inflammation, addresses intestinal spasm, and demonstrates laboratory activity against the most common gastric pathogen in the human population is not primarily a symbol, it is a prescription. Item 12. Garlic. Garlic occupies a different position in the gut health ranking than in the general healing ranking, because its gut-specific effects go well beyond its antimicrobial properties. Garlic contains fructooligosaccharides, the same prebiotic fibers found in leeks and onions, which selectively feed beneficial gut bacteria. But garlic's most gut-specific compound is allicin, and the mechanism of its gut action is more targeted than most people understand. A 2019 randomized controlled trial published in Frontiers in Physiology tested garlic extract supplementation on gut microbiome composition and found significant increases in lactobacillus populations and significant reductions in proteobacteria, a bacterial phylum that includes most of the pathogenic species associated with gut inflammation, intestinal permeability, and systemic immune activation. Proteobacteria bloom in dysbiotic microbiomes. Their overabundance is associated with inflammatory bowel disease, leaky gut syndrome, and, in recent research, with depression and cognitive decline through the gut-brain axis. Garlic supplementation consistently reduces proteobacteria populations in clinical studies. Additionally, allicin has documented specific inhibitory activity against Helicobacter pylori, the ulcer-causing pathogen we encountered with hyssop, with a 2016 meta-analysis finding consistent anti-H. pylori activity across multiple in vitro and animal studies. The ancient world ate garlic in quantities that would alarm most modern nutritionists. The microbiome evidence suggests they were right to do so. Item 11. Cumin. Cumin earned its place in the healing foods ranking through its metabolic evidence. In the gut health ranking, it belongs for a separate reason. Its carminative properties are among the most extensively documented of any culinary spice. Carminative herbs reduce intestinal gas production through two mechanisms. They inhibit the gas-producing fermentation of undigested carbohydrates by bacteria in the colon, and they relax the smooth muscle of the intestinal wall, facilitating gas transit and elimination. Cumin has been documented as a carminative in Ayurvedic texts, ancient Egyptian medical papyri, and Hippocratic writings simultaneously. 3 independent medical traditions assign cumin the same gut function. Modern clinical evidence confirms this. A 2013 randomized controlled trial published in the Middle East Journal of Digestive Diseases tested cumin extract against placebo in subjects with irritable bowel syndrome over 8 weeks. The cumin group showed significant improvements in bloating, intestinal cramping, and bowel regularity compared to placebo. Additionally, thymoquinone, cumin's primary active compound, has documented inhibitory effects on Candida albicans, the fungal organism that overgrows in dysbiotic gut environments and is associated with a range of systemic health complaints in individuals with compromised microbiome diversity. The ancient world consumed cumin at every meal across virtually every culture in the ancient Near East. Modern gastroenterology would describe that pattern as a daily anti-dysbiosis intervention. We are halfway through 21 foods, and I want to stop here for a moment. If what you have heard so far about butyrate from lentils, about pomegranate and urolithin conversion, about garlic reducing proteobacteria, has shifted how you think about what you eat and why, hit the like button. I want to be direct about why that matters. Gut health research is one of the fastest moving fields in biomedical science right now. The Stanford 2021 trial I referenced at the opening is genuinely one of the most important nutritional studies published in the last decade. And it received a fraction of the media coverage that a new weight loss supplement gets. That gap between what the peer-reviewed literature says and what reaches ordinary people is exactly what this channel exists to close. A like helps this video reach people who need this information. A subscription brings you back every week when I publish the next piece of research. The second half of this list is where the evidence becomes most striking. The foods from 10 through 1 are the ones that ancient physicians prescribed most specifically for gut complaints, and the ones that modern gastroenterology has validated most precisely. Let us continue. Item 10. Coriander seed. Coriander appears in Exodus, where the appearance of manna is compared to coriander seed, indicating that coriander was so familiar to the Israelites that it served as a visual reference point. It was not an exotic spice. It was a daily food. For gut health, coriander seed contains linalool, a monoterpene with documented antispasmodic effects on intestinal smooth muscle and established anxiolytic properties through the gut-brain axis. The gut-brain axis is the bidirectional communication pathway between the enteric nervous system, the 100 million neurons embedded in the gut wall, and the central nervous system. Approximately 90% of the body's serotonin is produced in the gut. The state of the gut microbiome directly influences serotonin production, which in turn influences mood, anxiety, and cognitive function. A 2012 randomized trial published in the Journal of Ethnopharmacology found coriander seed extract significantly reduced intestinal spasm and gas production in subjects with functional dyspepsia, the clinical term for chronic digestive discomfort without a structural cause. Additionally, coriander has documented heavy metal chelation properties. It binds to mercury, lead, and aluminum ions and facilitates their excretion through the gut. In populations with significant environmental metal exposure, this is a clinically meaningful detox pathway. The ancient world consumed coriander as a daily food. Modern toxicology has identified a heavy metal chelation mechanism in the same plant. Item 9. Bitter herbs. Bitter herbs are prescribed specifically in the Passover narrative in Exodus and Deuteronomy. They are not optional additions to the Passover meal. They are commanded. Horseradish, chicory, romaine lettuce, horehound. These were the bitter herbs of the ancient Near East, consumed in a ritual context that embedded a specific dietary practice into cultural observance. The clinical significance of bitter compounds for gut health is extensive and increasingly recognized in modern gastroenterology. Bitter compounds activate taste receptors not just on the tongue, but throughout the gastrointestinal tract. These receptors, TAS2Rs or type 2 taste receptors, are distributed across the entire gut lining, and their activation triggers a cascade of digestive responses: increased bile production by the liver, increased secretion of digestive enzymes by the pancreas, and stimulation of motility throughout the digestive tract. This is the mechanism behind the traditional practice of bitter aperitifs before meals across European and Mediterranean cultures. Amaro, bitters, gentian-based digestive drinks. They were not primarily alcoholic beverages. They were bitter herb preparations that activated the digestive system before eating. A 2018 review in Molecular Nutrition and Food Research analyzed the evidence for bitter compounds in gut health and found consistent evidence for improved bile flow, enhanced digestive enzyme activity, and improved gut motility across multiple bitter plant extracts. The Passover requirement to eat bitter herbs was a nutritional prescription embedded in religious practice. The ancient physicians who wrote those dietary laws understood, empirically, without the vocabulary of TAS2R receptor pharmacology, that bitter herbs prepared the digestive system for the meal that followed. Item 8. Goat milk and fermented dairy. The biblical description of the Promised Land as a land flowing with milk and honey is not poetry for abundance alone. Milk, specifically goat and sheep milk, the dominant dairy animals of the ancient Near East, was a central daily food source. But the milk of the ancient world was not pasteurized. It was raw, often naturally fermented through the action of ambient bacteria and the warmth of the climate. The clabbered milk, yogurt-like preparations, and fermented cheese of the ancient world were probiotic foods by default, containing live cultures of lactobacillus and other beneficial bacteria. The clinical evidence for fermented dairy on gut microbiome health is among the most extensive in microbiome research. A 2021 review in Nutrients analyzing 34 clinical trials on fermented dairy consumption found consistent increases in gut microbiome diversity, consistent reductions in Enterobacteriaceae, pathogenic bacteria, and consistent improvements in intestinal permeability markers across all trials. Goat milk specifically warrants mention because its protein structure differs from cow milk in clinically relevant ways. Goat milk contains predominantly A2 beta-casein protein rather than the A1 variant dominant in most commercial cow milk. A1 beta-casein is cleaved by digestive enzymes into beta-casomorphin 7, a peptide that has been shown in multiple studies to increase intestinal permeability and gut inflammation in sensitive individuals. Goat milk's A2 protein does not produce this metabolite. The ancient world consumed fermented goat and sheep milk daily. The Stanford 2021 study showed fermented dairy as a primary driver of the microbiome improvements in its treatment group. The ancient prescription and the modern clinical trial are pointing at the same food. Item 7. Soured grain bread, what we now call sourdough fermentation. This is the food most directly validated by the Stanford 2021 trial, and I want to spend the time it deserves. When scripture references leavened bread, the bread consumed daily throughout the year outside of Passover, it is referring to bread made through natural, slow fermentation by wild yeast and lactic acid bacteria. The process took many hours. The bread was slightly sour. The microbial community responsible for the leavening was complex and alive. This is sourdough, not the commercial product sold in most grocery stores today, which is frequently made with accelerated industrial processes and flavored with vinegar to simulate sourness. Real sourdough, slow fermentation by lactobacillus species and wild yeast, producing a fundamentally different food from modern commercial bread. The differences are clinically significant. During sourdough fermentation, lactic acid bacteria partially break down phytic acid, an anti-nutrient present in whole grain that binds minerals including iron, zinc, and magnesium, and prevents their absorption. The fermentation process increases mineral bioavailability by 30 to 50% compared to unfermented grain. Sourdough fermentation also partially breaks down gluten, the protein that causes immune reactions in celiac disease and intestinal inflammation in gluten-sensitive individuals. Multiple clinical studies have shown that subjects with non-celiac gluten sensitivity tolerate properly fermented sourdough bread significantly better than commercial bread made from the same grain. A 2019 randomized controlled trial published in Gastroenterology tested sourdough bread against commercial bread in subjects with irritable bowel syndrome. The sourdough group showed significant reductions in IBS symptom scores, bloating, and intestinal transit discomfort. The commercial bread group showed no improvement. The ancient world ate slow fermented grain bread as a dietary staple. The Stanford trial identified fermented foods as the primary driver of microbiome diversity improvements. The 2019 Gastroenterology trial showed sourdough specifically reducing IBS symptoms compared to commercial bread. The ancient bakers who allowed their dough to ferment slowly overnight were not following tradition for tradition's sake. They were producing a food that was, by every modern clinical measure, superior to the fast-risen product that replaced it. Item 6. Fermented fish preparations. This one surprises people, but it belongs on this list, and it belongs here. The ancient Near East produced and consumed fermented fish preparations extensively. Garum, a fermented fish sauce used across the Mediterranean world, was among the most commonly traded food commodities of the ancient world. Roman cooking used it in quantities that modern culinary historians find almost incomprehensible. Ancient Egyptian texts reference fermented fish preparations. They appear in the archaeological record of virtually every ancient Mediterranean and Near Eastern culture. These preparations were made by fermenting whole fish or fish viscera with salt, a process that takes weeks and produces a liquid intensely rich in short-chain fatty acids, amino acids, and beneficial bacteria. The microbiome relevance is twofold. First, fermented fish are a source of live lactobacillus cultures, the same beneficial bacteria as yogurt, sourdough, and other fermented foods. Second, fermented fish are an exceptionally concentrated source of long-chain omega-3 fatty acids, EPA and DHA, which have documented anti-inflammatory effects on the gut lining and established roles in maintaining intestinal barrier integrity. A 2020 randomized controlled trial found that omega-3 supplementation significantly improved intestinal barrier function, measured by lactulose-mannitol ratio, the standard clinical test for intestinal permeability, in subjects with inflammatory bowel disease. The ancient world consumed fermented fish preparations daily. They were getting probiotic cultures, short-chain fatty acids, and omega-3s simultaneously from a single food source. Modern functional food science would struggle to engineer a more comprehensive gut support product. Item 5. Vinegar. Vinegar appears in the Book of Ruth. Boaz invites Ruth to dip her bread in vinegar during the harvest meal. Vinegar is referenced in the Psalms. Proverbs mentions vinegar in the context of its sharp, penetrating character. In the ancient world, vinegar was a daily condiment and a medicine. The active component of vinegar is acetic acid. And acetic acid is the same short-chain fatty acid produced by gut bacteria during the fermentation of prebiotic fibers. When you consume vinegar, you are delivering acetic acid directly to the gut environment. The clinical evidence for apple cider vinegar and wine vinegar on metabolic and gut health has become one of the more active areas of nutritional research in recent years. A 2019 randomized controlled trial published in the Journal of Functional Foods found that daily apple cider vinegar consumption over 12 weeks produced significant reductions in fasting blood glucose, HbA1c, and triglycerides in type 2 diabetic subjects, without any other dietary change. The mechanism involves acetic acid's inhibition of alpha amylase and sucrase, the enzymes that break down starch and sucrose in the gut, slowing glucose absorption. For gut health specifically, acetic acid has documented antimicrobial activity against pathogenic bacteria in the gut, including E. coli and Staphylococcus species, while appearing to spare beneficial lactobacillus populations. It also stimulates the production of mucin, the gel-like substance that forms the protective mucus layer coating the intestinal epithelium. The mucus layer is the gut's first line of defense. Its degradation is one of the early events in inflammatory bowel disease. Vinegar, consumed daily as the ancient world used it, may support the regeneration of that protective layer. Item 4. Olive oil. We established olive oil's cardiovascular and anti-inflammatory credentials in the last video. For gut health, olive oil occupies a distinct and underappreciated role. Extra virgin olive oil is the richest dietary source of oleocanthal, the ibuprofen-like anti-inflammatory compound we discussed previously. In the gut context, oleocanthal COX inhibiting activity directly reduces prostaglandin production in the intestinal wall, and excess prostaglandin production in the gut is a primary driver of the cramping inflammation and motility dysregulation that characterizes irritable bowel syndrome and inflammatory bowel disease. But olive oil's gut-specific mechanism goes further. A 2019 review in Nutrients analyzed the specific effects of extra virgin olive oil polyphenols, including oleocanthal, oleuropein, and hydroxytyrosol, on gut microbiome composition. The findings: consistent prebiotic-like effects, selectively feeding lactobacillus and bifidobacterium species, and consistent reductions in potentially pathogenic Clostridiales species. Additionally, olive oil's fat content, high in monounsaturated oleic acid, specifically stimulates the production of GLP-1, a gut hormone that regulates intestinal motility, reduces intestinal inflammation, and signals satiety to the brain. GLP-1 is the same hormone targeted by the new class of GLP-1 receptor agonist drugs being prescribed for diabetes and obesity. Olive oil stimulates natural GLP-1 production. Pharmaceutical companies sell synthetic GLP-1 analogs for hundreds of dollars per month. the ancient world consumed the food that stimulates the endogenous hormone daily as a cooking fat, a condiment, and a medicine. Item 3. Fermented garlic. Specifically, black garlic and aged garlic extract. I want to distinguish this from raw garlic, which appeared at item 12. Ancient populations did not consume garlic only raw. They stored it, allowed it to age, fermented it in salt brine preparations, and used aged preparations medicinally. The fermentation of garlic transforms its chemistry in clinically significant ways. Raw garlic's primary compound is allicin, which is potent but unstable and irritating to the gut lining in large amounts. Fermented and aged garlic converts allicin into a range of stable compounds, S-allylcysteine, S-allylmercaptocysteine, and others, that retain antimicrobial and anti-inflammatory activity while being significantly better tolerated by the gut epithelium. A 2019 randomized controlled trial specifically examining aged garlic extract, the fermented form, found significant increases in lactobacillus and bifidobacterium populations, significant reductions in Clostridia, and improvements in self-reported digestive symptoms compared to placebo over 12 weeks. The gut-specific finding that elevates fermented garlic to number 3 is this: aged garlic extract has documented stimulatory effects on the production of mucin, the protective gut mucus layer, in intestinal epithelial cells. The same mechanism we identified for vinegar, but at a different point in the mucus production pathway. Two different ancient foods, two different mechanisms, both targeting the same essential protective structure of the gut. The ancient world consumed both daily in combination. The clinical evidence suggests this combination was not coincidental. Item 2. Fermented grain beverages. What the ancient world produced from soured grain, and what modern fermentation science recognizes as among the most complex probiotic foods available. The ancient world did not only ferment its solid foods, it fermented its beverages. Kvass, a fermented beverage made from bread, grain, or beet, has been produced across Eastern European and Middle Eastern cultures for thousands of years. Ancient Egyptian workers received fermented barley beer as a daily ration. Ancient Near Eastern texts describe fermented grain beverages in contexts that make clear they were consumed for health, not purely for intoxication. The distinction between the fermented grain beverages of the ancient world and modern commercial beer is critical. Ancient fermented beverages were low alcohol, short fermented, and consumed fresh, retaining live bacterial cultures, unfermented prebiotic fibers, and a full range of fermentation metabolites. Modern commercial beer is pasteurized, filtered, and produced through industrial processes that eliminate the probiotic content entirely. What the ancient world consumed was functionally closer to a probiotic drink, rich in lactobacillus species, prebiotic oligosaccharides from the grain, short-chain fatty acids from fermentation, and B vitamins produced by the bacterial metabolism. A 2020 clinical study examining traditional kvass fermented for 48 to 72 hours from whole grain found it contained viable Lactobacillus plantarum, counts comparable to commercial probiotic supplements, along with measurable concentrations of butyrate acetate and propionate, the short-chain fatty acids associated with gut barrier integrity and anti-inflammatory signaling. The Stanford 2021 trial showed that high-fermented food diets outperform high-fiber diets on gut microbiome diversity and inflammatory markers. The ancient world was consuming fermented grain beverages daily. The Stanford trial was, without intending to, validating a 3,000-year-old dietary practice that has been largely abandoned by modern Western populations. Item 1. This is where I want to do something different from the standard format. Number 1 on this list is not a single food. It is what happens when you consume these 21 foods together in combination as a daily dietary pattern, which is exactly how the ancient world consumed them. Let me explain why this matters clinically. The Stanford 2021 trial did not test individual fermented foods in isolation. It tested a dietary pattern, a cluster of fermented foods consumed consistently across daily meals. The improvements in microbiome diversity and reductions in inflammatory proteins observed in that trial emerged from the combination, not from any single food. This reflects a principle that modern nutritional research is increasingly recognizing. The gut microbiome responds to dietary patterns, not to individual nutrients. The diversity of fiber types, prebiotic compounds, probiotic organisms, polyphenols, and fermentation metabolites consumed across an entire diet shapes the microbiome more powerfully than any individual supplement. The ancient biblical dietary pattern consumed these foods in specific combinations at specific times. Through specific preparation methods, soured grain bread fermented slowly overnight. Fermented dairy, yogurt, and aged cheese, consumed with meals. Raw garlic and onions eaten daily, providing prebiotic fibers. Lentils cooked and cooled, maximizing resistant starch and butyrate production. Bitter herbs consumed before or with meals, activating digestive enzyme secretion. Vinegar used as a condiment and food preservative. Olive oil as the primary cooking and dressing fat. Fermented grain beverages consumed in moderate amounts throughout the day. This was not a designed protocol. It was a practical dietary pattern shaped by the agricultural, culinary, and preservation technologies of the ancient Near East. But when you map it against the current microbiome literature, against the Stanford trial, against the Cochrane reviews on prebiotic fibers, against the clinical evidence for fermented foods, against the research on short-chain fatty acids and gut barrier integrity, you find something remarkable. The ancient dietary pattern hits every major lever of microbiome health simultaneously. It delivers live probiotic organisms from fermented dairy and fermented grain. It delivers prebiotic fibers from lentils, onions, leeks, and figs that feed those organisms. It delivers polyphenols from pomegranate, garlic, and olive oil that selectively suppress pathogenic bacteria. It delivers short-chain fatty acids from fermented grain beverages and resistant starch that fuel the colonocytes lining the gut. And it delivers the bitter compounds from hyssop and bitter herbs that activate the digestive enzyme cascade that makes all of this absorption possible. The ancient world did not design this. They did not have the vocabulary of prebiotics, probiotics, short-chain fatty acids, or tight junction proteins. They had generations of empirical observation. The accumulated clinical data of millions of people eating these foods over thousands of years, and a culture that embedded these dietary practices in daily life so thoroughly that they were recorded in the texts that have survived to this day. The Stanford trial validated that pattern, not intentionally, not with ancient texts as the reference, but with randomized controls, blood work, and fecal microbiome sequencing. The conclusion was the same. The people who ate the fermented, fiber-rich, polyphenol-dense dietary pattern that the ancient world consumed daily had healthier guts, more diverse microbiomes, and lower systemic inflammation than the people eating the modern high-fiber diet. The ancient table was not behind us. In terms of gut health, it was ahead. 21 foods, 3,000 years of daily consumption. One 2021 clinical trial that confirmed what the ancient world had worked out by observation. I want to be honest with you about the limits of what I have covered today. Not every claim in ancient medicine is validated by modern research. The ancient world also prescribed foods and practices that turned out to be wrong, harmful, or simply ineffective. I am not arguing for uncritical deference to ancient authority. What I am arguing is this: when multiple ancient medical traditions, across different cultures, across different centuries, with no communication between them, converge on the same dietary practices, and when modern clinical research then runs the trials and finds measurable biological effects, that convergence deserves serious attention. The gut microbiome is not a wellness trend. It is the foundational regulatory system of human health. The ancient world ate in ways that kept it diverse, robust, and functional. Modern Western dietary patterns, characterized by ultra-processed food, antibiotic exposure, low fermented food consumption, and inadequate fiber diversity, have produced what researchers now call the industrialized microbiome, less diverse, less resilient, and more associated with chronic disease than the ancestral microbiome it replaced. The 21 foods on this list are not exotic. They are not expensive. They are not locked behind a prescription or a clinical protocol. Most of them are available at your grocery store today. The question is not whether the evidence for them is real. It is. The question is whether you are willing to eat the way the ancient world ate, not for religious reasons, not for cultural nostalgia, but because the clinical literature keeps arriving at the conclusion that they were eating correctly for gut health in ways that modern nutrition is only now beginning to fully understand. If this video shifted how you think about gut health, if the connection between what the ancient world ate and what the Stanford trial found makes you want to change something on your plate this week, hit the like button. And then do one thing right now. Subscribe to Covenant Table and turn on the notification bell. Because next week, I am going to cover something I referenced today but did not fully unpack: the gut-brain axis. The specific pathway by which your gut microbiome influences your mood, your anxiety levels, and your cognitive function. And I am going to map the ancient biblical foods that specifically support that pathway against the current neuroscience research. The ancient world did not separate gut health from mental health. They treated them as the same system. Modern neurogastroenterology is arriving at the same conclusion. Before I go, I want to ask you a specific question for the comments section, and I want you to answer honestly. How many of the 21 foods I covered today do you currently eat on a regular basis? Not occasionally, regularly, weekly. Drop the number in the comments. And if the number is lower than you expected, that is not a criticism. That is useful information. And it is exactly the kind of data that shapes what I cover on this channel. I read every comment. I will see you next week.